In the past century, the recombinant DNA technology was just an imagination that desirable characteristics can be improved in the living bodies by controlling the expressions of target genes. However, in recent era, this field has demonstrated unique impacts in bringing advancement in human life. So, I'm going to start by explaining what is biotechnology. It is the use of microorganisms, cells, or cell components to make a product. This product can be foods, antibiotics, vitamins, or enzymes. Recombinant DNA technology, it's a type of biotechnology. And recombinant DNA is the DNA that has been artificially manipulated to combine genes from two different sources. These two different sources are from unrelated species and are really different. Recombinant DNA does not occur in nature and is only done in vitro. Now, an overview of recombinant DNA. First, we get a plasmid from bacteria, and the cool thing about plasmid is that it can divide on its own. We use this plasmid from bacteria and get a restriction enzymes, and these restriction enzymes will find its restriction site and break its sugar phosphate backbone, exposing its sticky ends. Then we insert genes from other sources into the plasmid by DNA ligase. DNA ligase will stick the complementary sticky ends. Then we insert this modified plasmid into a vector. We can have two objectives or two goals. It can be to produce more copies of a gene or to produce proteins or protein products of the gene. Now, tools for genetic engineering, restriction enzymes are needed, vectors, even DNA ligase are needed in genetic engineering. So I'm going to start by talking about restriction enzymes. Restriction enzymes are naturally produced by bacteria, and they are used to destroy bacteriophage DNA in bacterial cells if they were ever attacked. And restriction enzymes have two ways of cutting, and by cutting I mean breaking down the backbone of or breaking down bones between nucleotides. Restriction enzymes have two ways, as I said. It's creating sticky ends or creating blunt ends. Now, ligase, it's like the glue that sticks complementary strands of DNA, and it's really important in everything basically, and it needs ATP, it needs energy. DNA ligase has extensive use in molecular biology laboratories for genetic recombinant experiments. Now, vectors are small pieces of DNA used for cloning, and the gene to be inserted into the genetically modified organism must be combined with other genetic elements in order for it to work properly. Now, the requirements of the vector, it should have self-replication, a cloning site, a promoter, and selectable marker, antibiotic resistance, and proper size. Now, here's a plasmid with its own restriction sites. Host for a DNA recombinant technology can either be bacteria, yeasts, plant cells, or mammalian cells. What they all have in common is that they are easily grown in vitro. Except for mammalian cells, they're a bit harder than bacteria and yeast and plant cells, of course, because they are more complicated. Now, this technology has faced a lot of problems, and not all people are supportive. The future of genetic engineering is contained in both an awesome yet uneasy picture. There are many moral issues involved with genetic engineering in all areas. Huge numbers of people are both against and supportive of human cloning or changing the genetic code or the genome or the use of recombinant DNA. And some believe that it's unethical and unfair. It can have also religious problems too. But even though scientists, even though it does not have a lot of supporters and it has a lot of accusations, scientists predict that the future of genetic engineering is very bright and it will occur soon. The future is unknown in specifics, yet the dreams and hope of scientists and common society soar far ahead in time. With dedicated research, inspired scientists, and hard work, what might be set for ahead of us could be brought to us sooner.